Welcome to the live webinar, Achieving R&D Goals with Fast, Fresh, On-Demand Biospecimens Collected at Your Lab or Office.
And thank you all for attending. So as Mega had just noted, to give a high-level overview of the marketplace and some of the things that we have seen. In the marketplace, as I'm sure you're all aware, there are many ways to procure patient samples, whether that be blood banks or inventory, physicians or academia relationships. But some of the challenges we've heard from our clients who utilize these different methods are they really struggle to find the patients that they are looking for with their specific IE criteria. And they're not always getting the data they need about these donors. There could be sample variability because those samples are not being collected in the right method, or they're not getting enough volume from those samples. To really solve some of these problems, or as we here at Sangwin like to say, bridge the translational gap, Sangwin has developed a direct-to-patient model where we interface directly with patients and donors to find and deliver the specific biospecimens that researchers are looking for. Sangwin's product set has expanded so much over the years, as has our community. We have over 70,000 patients and healthy donors within our community that have signed up directly to participate in research through our relationships with patient advocacy organizations, digital marketing, and social media, which really gives us direct access to medical records, patient-reported outcomes, and demographics. And because of this, we can collect and process minimally invasive samples through our fleet of mobile phlebotomists all across the United States, and leukopaks through our apheresis sites all across the US, and our lab in San Diego to help with that processing. But one of our staple offerings has remained our on-site programs, which we will be focusing on today for this webinar. In our experience with on-site programs, we have recruited over 700 donors across 18 on-site programs, and I'm happy to report that number continues to climb. We've achieved over a 90% fulfillment rate for all of these studies, and we've been successfully running these programs for almost 10 years. Many of our clients renew year after year. One of our top clients has been going strong since 2016. And we have a specific dedicated staff to work on these programs, including our on-site specialist, Megha Patel, who will be helping with the Q&A at the end of this webinar today. We have clients come to us to launch these programs for a variety of reasons. But one of the top reasons always being the need for fresh samples, whether that be for single cell assays, immunological assays. The ability to get them within minutes of collections allow researchers to enable the best science for their experiments. Other reasons can include control samples for other projects or studies that truly need a fast turnaround time from the request to delivery. Some researchers don't know weeks in advance when they will need samples, so being able to have a quick turnaround time and flexibility is really helpful for them to perform the experiments they need, when they need, and how they need them. And then, of course, on the employer side, this program is a great way to engage employees to participate in the R&D for the organization, which can, of course, also result in savings. The reliability and knowledge of handling these samples reduce variability, so you may need fewer samples to get that high quality data for your experiments. And of course, the fast turnaround, fast and speed always saves money as well. So what does your company need to do to launch a program like this? One of the things that we hear so often is, yes, we have an on-site program, or we've had one in the past, but we had to handle everything internally. And the biggest concerns are trained on internal resources and abiding by privacy policies, specifically the blinding and anonymization of participants, creating SOPs for all of these policies, and developing a privacy policy. By utilizing internal resources, whether that be for the phlebotomy or for the screening, consenting and scheduling, there is a higher risk of non-compliance because these employees are unblinded to the participants and their samples. But with Sangwin, those aren't things you need to worry about. So for these programs, we identify and retain a reliable phlebotomist to collect all minimally invasive sample types. That includes blood, saliva, urine, certain swabs, skin capes, etc. We create and submit a custom IRB protocol and manage all regulatory requirements. We pre-screen and consent all of the interested donors, while most importantly, managing the HIPAA compliance and the privacy. So what does your organization need to do to participate in this program? All you truly need to do is provide a room. So as long as you dedicate a collection room for these collections to occur, that usually includes, a chair, some supply storage in close proximity to a sink, then we can run a program like this in your facility. Our experience in these programs is to really try and make it as convenient, flexible, and customizable as possible for both the researchers and the donors. So we'll dive into this process just a little bit more. So at Sanguine, we built out custom forms and a landing page for your organization to put in these blood requests as few as 3 to 5 days in advance. That way, everyone knows how easy it is to sign up to be a donor, as well as put in the request forms for the specific samples they need. The donation time is only 15 minutes long, so it's a really quick break from the workday and convenient for donors to continue about their day. Once the donation is complete and the donor has left the area, the phlebotomist will notify the researcher that the blood is ready and provide the pickup location for them to instantly get their blood, ensuring the privacy that the donor and the researcher aren't passing in the halls. I had mentioned Sanguine is an experienced provider. So we handle all of the writing and the submission for the IRB protocol. We can usually get this program up and running within 6 weeks. We, of course, make sure that our phlebotomists are certified. So you can see on the screen here, the various certifications they go through to become a phlebotomist with Sanguine. We even complete program-specific training so they understand all the requirements and nuances to work within your organization. We work with you to define those specific days and times for the phlebotomist to be at your facility, ready to do those draws, so that way researchers can plan accordingly, knowing that the phlebotomist may be there on Tuesdays or on Thursdays at specific times so they can plan their experiments. And of course, we understand life happens. People may get sick or someone's car might break down. So we also try and have a backup phlebotomist in the area to step in when needed. And we also try and manage backup donors to try and limit any reschedules. So that way researchers always get their blood when they are depending on it. We have a whole team dedicated to making sure that your on-site program runs smoothly. We have dedicated research consultants who handle the scheduling and the receiving of requests, even ones that are repeat donor requests. And we also have them manage a company-specific on-site email address where someone on our side will be able to quickly answer and accommodate any flexibility for requests, concerns, changes. And you'll also have a dedicated project manager to make sure that everything's running as it should. If you run this program internally, you may have to dedicate several employees to assist with that day-to-day, to do even things such as managing the blood volume for repeat donations, making sure they don't go over that specific threshold based on the protocol, consenting and scheduling donors, managing the phlebotomist hours and supply management for all of the specific tubes you need, IRB and IRS compliance, those privacy policies and SOPs I had mentioned, as well as just fielding those real-time issues that require immediate attention for successful collections. So by utilizing Sanguine's experienced team, you can really free up those resources and not have to worry about the program's success. Next, one of the major components of our program is flexibility. You really have the ability to specify any specific inclusion and exclusion criteria. So for example, if you're only looking for females or for males or someone of a certain age, even smokers or high caffeine users like myself, we really have the ability to screen that and find those patients based on your specific requests. We even got a request the other day who said, we really need to make sure our donor on this day doesn't have NSAIDs within, the past 48 hours. We always like to try and accommodate those specific requests for you. We also handle all of the collection specific requirements. When we're preparing for your onsite, we identify which tubes you would like us to provide, so that way we are prepared for all of your visits. But then it also gives the researchers flexibility if they want to switch up their tube type or the volume. For example, one day they might want sodium heparin for 10 mLs. Another day they might want sodium citrate for 50 mLs. We are able to accommodate those changes based on what researchers need for those studies. Another thing that we can do is recall specific donors or even design longitudinal studies. For the majority of our on-site programs, we have an 80% retention rate for all of our donors. And what we do is we provide them a specific PID, is what we call it, patient identification number, to make it easy for you to call your project manager and say, hey, this sample worked out great for me a few weeks ago. I'd really like to schedule another donation from PID 1, 2, 3, 4, 5, and 6, 7, 8, 9, 10, while still keeping that donor anonymous. We can manage all of the logistics and requests, and as I mentioned, really freeing up those internal resources and leaving all of those details to us. And by having that custom protocol, it really does allow you to design the program to meet all of your organization's needs. So how do we find people to participate? Of course, we need to find the donors. So to make that easy to get the word out to all of your employees about this program, hey, this exists, Sanguine actually builds out custom marketing material for you to share with all of the employees in that location, similar to one you see here. In addition to that, we have an onboarding meeting for both interested participants as well as interested researchers to really explain the nuances of the program, so that way they understand, how can I donate or how can I request blood throughout this. We even collect a custom landing page to make it easy for donors to sign up and share all of the pertinent information to participate, including custom donor compensation for their participation, an overview of the screening process and the informed consent forms, explaining how appointments are scheduled and set up, to really make it easy for them to understand, hey, what does this process look like? I had someone tell me the other day that, yeah, we have this program, but we actually didn't know how to sign up, or we didn't know where these resources were. And then they found it was just an email that they had to dig down deep into the archives to find. Well, we make it easy by having all of this information ready on that landing page, easy to make available to all of your employees. So once a donor signs up on the landing page, they're contacted by a Sanguine employee to provide them with an overview and obtain signatures for the informed consent to participate. From there, they conduct a phone screening to determine if they are eligible to participate. And if so, they may be scheduled for an upcoming collection in which they'll receive an email with the donation time and location and the phlebotomist name, or they'll be contacted at a later date for future donations. On the researcher side, after we complete that orientation, requests can be submitted by the custom request form that we will create for you. And from there, the donors will get screened and scheduled, as we just saw, and the researcher will get an email confirmation with the de-identified sample collection details. And when the sample is ready for pickup, the phlebotomist will notify the researcher via email or phone, and with the sample pickup time and location instructions. So as a reminder, we have that different sample pickup location to really ensure that privacy. So as you can see here, it's a very easy process from both sides, the researcher and the donors. There we go. So you've seen that we've provided several companies with ongoing on-site programs. And some of the science those collections contributed to have actually been published in peer-reviewed journals. So here are a few examples to give you an idea of the breadth of the R&D questions these specimens can address, including drug development assays, healthy controls for discovery work, biomarker development. But I will walk you through one specific published case study that we have. So this first study from one of the teams at Gilead Sciences aimed to evaluate the repercussions of delaying the processing of PBMCs after collection. PBMCs are an increasingly popular sample type for identifying and tracking biomarkers in an immune-related therapeutic development. But the viability of these cells depends on how they were handled. Since the whole blood collected from their on-site program was available to them within minutes of collection, they could delay subsequent PBMC isolation from any amount of time they wanted. And then they conducted a host of analytical methods typical of a clinical study involving PBMCs to really track the damage associated with the delay. In this case, the first time point was just 2 hours post-collection. Looking at several donors, they observed that the PBMC viability was reduced after 48 hours. Not a big deal. You can get away with the processing delays up to 2 days. Well, then flow cytometry told them they were getting granulocyte and neutrophil contamination within 24 hours of processing delay in most donors. That brings it down to about 1 day. And next, they looked at the single-cell RNA sequencing data and found gene expression profiles for inflammatory proliferation and signaling pathways were all increased, but the IFN gamma and metabolic pathway gene expression profiles were decreased. So overall, they were seeing gene expression correlations in under 0.8 after a 24-hour delay, meaning the processing delay was fundamentally changing how T cells, B cells, and K cells, monocytes, and dendritic cells were behaving. In other words, the cell's underlying biology was fundamentally different by 24 hours. They looked at the IFN gamma secreting cells, specifically with ELISA assays, and found that their numbers were reduced with longer delays. And you can see that while the decreasing pattern is similar, the extent of the decrease changes from donor to donor. So taken together across these methods, delays in the PBMC processing changed the underlying biology of the donor immune cells, and those cells were donor dependent. So that's a lot of variability in downstream assays, which means less useful results and higher costs in recruiting more patients to trials. So the team recommended minimizing those delays in clinical trials by having processing capabilities nearby at the clinical site itself if possible. So by having an on-site program enabled this team to access these study samples on demand and design exquisitely the processing delay time of experiments, allowing them to pinpoint exactly when trouble started for the PBMCs. We do offer PBMC processing at our San Diego lab if that's also of use, but to bring it back to our on-site program, the biggest takeaways we want to have are that Sangwin's on-site program really combines convenience, experience, and flexibility to provide you with an end-to-end solution for fresh sample needs. So that concludes the presentation portion, but we will hand it back over to Megha to answer some Q&A.
Thank you, Chelsea, for that informative presentation. I hope you all feel the same way too. Now it's time for our Q&A session. Please use the Q&A feature to submit your questions. I see a few interesting ones already. So our first question is, what sample types can you collect?
That's a good one for the first one. The different sample types that we can collect for these programs are typically whole blood. So that is the most common request that we receive, but we can get other minimally invasive sample types. For on-site programs, the most common ones that we see are skin taping. So it wouldn't be it wouldn't be a puncture, just that skin taping of the top of the skin, hair, Yes, I think we have done some eyebrow ones. So if anyone needs an eyebrow trim during the workday, that can help, as well as other minimally invasive samples. So saliva, urine, RNA, DNA. But since we are doing custom protocols, if there are other sample types, we can always build those in as well.
Great. Second question is, will I be updated regarding the status of my sample request?
Great. Mega, you want to take that one?
Absolutely. So absolutely, you will be notified along the request process. So as soon as you submit through a customized sample request form that Chelsea had mentioned earlier, you would receive an email alert just letting you know that we've received your request and that we're currently working on scheduling out your donors. And then as soon as we've scheduled out all of your confirmed donors, you would also receive a request at that point, or sorry, an alert at that point. And then once all of your samples have been dropped off and they're ready for pickup, kind of just limiting the back and forth and having you wonder, when are my samples going to be delivered? Should I leave my lab now? Just making it really easy for you to be kept in the loop the entire way.
Third question is, what if I don't feel comfortable donating the full 180 ml volume as a donor? It's a good one.
Mega, do you want to work that one through?
Yeah, of course. So that's absolutely not a problem. If you know that you have a history of fainting or anything like that over a certain amount, you just don't feel comfortable, you can just let our dedicated study coordinator know, and we'll note that down internally in our system and just make sure that you're not scheduled for any donations above that amount.
All right, I see a few more. Are you able to process plasma serum samples from whole blood at the site before shipment?
Yes, so Mega, do you know if we can do centrifugation capabilities on site if we equip that with our phlebotomist?
Absolutely. So all of our phlebotomists are trained on centrifugation in home, so we could certainly bring that logistically on site as well. It would probably just maybe increase the amount of time it would take, but it's definitely a possibility for sure.
How many donors can we request a day, per day or per week, and how much blood can we request per donor?
Really good one. So for healthy donors, we can obtain up to 180 mLs within a 6-week period. In regards to how many you can request a day or a week, that's something that we plan out with you during the study design period to really determine how long you want a phlebotomist there, whether that's, some on-site programs, we have a phlebotomist on site multiple days a week for a specific amount of hours, whereas other smaller programs might just have someone there for one day for a couple hours, weekly or bi-weekly. So it really depends on how much volume you need from collections.
How long is a typical lead time approximately to set up on-site blood donation program, including ARB time and everything?
Yeah, really good one. So typically, since we are setting up the IRB protocol, we would say it's typically around a 6-week setup time.
Are you able to send out a portion of the collected samples to a third party for characterization, like Quest LabCorp?
Yes, we certainly could do that. So we would just build in specific shipments to send those out specifically. Mega, do you have any other comments about that?
Nope, I think what you said is accurate. We would just build that into your specific contract and just make sure that we have capabilities of drawing the blood and, driving it to an appropriate courier service or whatever it may be to get that offsite.
Great. A lot of questions, a lot of interesting ones. The next one is, are subjects screened for blood-borne pathogen? If not, what is the risk mitigation Sanguine have in place to ensure potentially infectious samples are handled correctly?
Great question. So typically we do screen the patient for a self-report for any infectious diseases. And if those are reported positive, then we would exclude that donor from participating. We have had some recent requests to do some of the rapid screening as kind of like a pre-visit in order to qualify a donor. So that is something that we could also consider if you really do need to take that extra step to get that testing.
Will Sanguine handle continual recruitment of new donors for the program?
Yeah, Mega, can you touch upon that?
Sure. So, like Chelsea mentioned earlier, we do build out that custom recruitment for you, and those flyers are available for you to circulate amongst your employee database as you please. So typically, you'll have an initial recruitment period, and then following that, every few months, we do ask you to just circulate recruitment by just circulating that flyer, maybe send out an email blast, just because we obviously don't have contact with your entire employee database. But as soon as you send out those flyers, we take care of everything after that from the screening and consenting aspect of it.
Next one, can a donor receive test results or do those remain confidential? And does Sanguine control the data, or does the company need to have a PI and a study coordinator too?
Mega, you want to take that one?
Yeah, sorry, just rereading this question. So in terms of test results, if we were to do any sort of maybe offsite testing or anything like that, or maybe you're using those samples on your own to do any testing, we would not be receiving any of those results. You would keep them confidential amongst whatever your, experimental purposes are. And then we do have a dedicated study coordinator that handles just the screening consenting portion. Other than, basic demographic information, those consent forms, they aren't privy to any other health information that the participant may, have. Does that answer the question? If not, you can provide additional questions off of that.
Perfect. Next one is, do you collect disease samples or just healthy normals? And if so, is it possible to get information from medical records? And are all those conditions self-reported?
Yeah, great question. For the on-site program, typically we stay with healthy controls. However, within our translational business, that is where we do collect disease samples based on your request. And in that case, we do get medical records for them to confirm that diagnosis. But for on-site programs, typically we are mainly dealing with healthy individuals for this program.
Do employees get compensated, and who sets that compensation?
Great. Yes, so we do recommend compensating the employees who donate for their time. In regards to what that compensation is, we typically give a recommendation. Usually we recommend about $100 per visit. However, that is up to you if you want to change that specific amount. And we would handle all of that for you. So we would provide a rechargeable gift card that they would receive at the end of the collection. Really good question.
Next one is, can sample be extended to non-employee donors if set up in a company?
Yeah, Mega, I think we have done this for for a couple different on-sites where, for example, they wanted to utilize, other folks within their building. We would just have to make sure that in the beginning, when we are planning out that on-site program, that we have a plan to include, expanding out of your organization to really make sure that, we're covered from the IRB protocol. I don't know if you have any other comments on that, Mega.
No, that's just it. As long as it's included in the protocol that, it's whatever company you are and your affiliates within the building, and you have approval from those other companies that their employees can participate with us, then it wouldn't be a problem at all.
Another question, do you have established relationships with third-party clinical labs that could be used for various sample characterizations?
So similar to if we did need to do additional testing or ship that out, that is something that we could do. We do have specific labs that we work with that could provide specific tests that you need. So again, that's something we would just want to know what those tests are to make sure that they're available. And then of course, plan out when we would need to ship those out. So that way we could build out your contract accordingly.
All right, and probably the last question of the session, how would the blighting work in a small company?
Yeah. So that's a good one because, people always ask us, how does it work? Does it matter what size our company is? So I know Mega can probably touch on this a little bit better than myself, but we like to consider a few different elements when working with smaller companies. I think one of the pieces is, how many collections and samples are you going to need over the course of the year? And then, of course, understand, how many employees do you have? Another element is we would really want to make sure that the room we would provide for collections is very private to make sure that we aren't having, one donor walk in next door and then, another go get it a couple seconds later if it's just one giant room, right? So we would really want to make sure that we have that privacy. Mega, I know you've worked with some smaller on-sites as well. Do you want to add any additional color to that?
Yeah, I think you touched on pretty much everything that I would have emphasized. But one of the big things is that because Sanguine is handling all of the screening, consenting, kind of all the RB portions, we are the only ones really that know who the donors are and what exact PID correlates. So on your end, when you actually get that sample, the only real information you have on the donor is that de-identified PID. And like Chelsea said, as long as the collection room and the drop-off location and pickup location for those samples is, fairly away from each other, that there's no way that anyone's going to cross paths, it's worked fairly well for our smaller companies, just as well as our large ones.